DNA transcription is a fundamental process in biology where genetic information encoded in DNA is converted into RNA molecules. This process is essential for gene expression, which ultimately dictates the synthesis of proteins and the regulation of various cellular processes. Initiation. Transcription begins with the binding of an enzyme called RNA polymerase to a specific region of DNA called the promoter. The promoter sequence signals the start of a gene and provides the necessary information for RNA polymerase to recognize and bind to the DNA. Elongation. Once RNA polymerase binds to the promoter, it unwinds the DNA double helix and begins to read the DNA template strand. Using one strand of DNA as a template, RNA polymerase synthesizes a complementary RNA molecule by adding nucleotides one by one. The nucleotides are selected based on the complementary base pairing rules, A with U in RNA and G with C. Termination. Transcription continues until RNA polymerase reaches a specific termination sequence in the DNA, signaling the end of the gene. In prokaryotes, termination signals often involve the formation of hairpin loops in the RNA transcript, causing the RNA polymerase to detach from the DNA template. In eukaryotes, termination is more complex and involves the recognition of specific termination sequences and the release of RNA polymerase from the DNA template. RNA processing in eukaryotes. In eukaryotic cells, the newly synthesized RNA molecule, called pre-mRNA, undergoes several processing steps before it can be translated into a protein. These include the addition of a 5' and a poly-A tail, as well as the removal of introns through RNA splicing, leaving only the exons, which contain the protein coding sequences. Overall, DNA transcription is a tightly regulated process that plays a critical role in gene expression and the synthesis of proteins necessary for cellular functions. Understanding the intricacies of transcription is essential for medical students to grasp the fundamentals of genetics, gene regulation, and molecular biology, which are crucial for understanding diseases and developing new therapeutic strategies.